Good day. I'd like to call the federal officer for NVAC. There are a few things you should know before we get started today. First, this is a public meeting and it's being recorded. All statements made on the today are made on the record. Second, this advisory committee is governed by the Federal Advisory Committee Act, or FACA for short. FACA provides rules about the circumstances by which agencies or officers of the federal government can establish or control committees or groups like this one to obtain advice or recommendations. The voting members are special government employees and are therefore subject to conflict of interest laws and regulations, as well as all of the members who work for the federal government. These members previously provided information about their personal, professional, and financial interests. Each voting member's financial interest and outside affiliation has been carefully screened, and we do this each year to ensure that they comply with federal ethics law. The liaison representatives are non-voting members of the advisory council and are not subject to the same FACA rules as the voting members. Additionally, the information provided at this meeting does not necessarily represent the official position of the National Vaccine Advisory Committee or the U.S. Department of Health and Human Services. Mention of products, processes, services, manufacturers, companies, or trademarks does not constitute its endorsement or recommendation by the U.S. government, the Department of Health and Human Services, or NVAC. And so with that, I will go ahead and turn it over to our wonderful chair, Dr. Bob Hopkins, to get us started.
All right. Good afternoon or morning, wherever you're calling in from, and welcome to this second day of our February 2024 NVAC meeting. I want to thank you, Anne, for getting us started, and to the NVAC team for its work in planning this meeting and supporting the committee throughout the year. I also want to express my appreciation to the NVAC members and subcommittee members for their dedication to further the work of this committee. I look forward to the presentations and discussions today. I'm going to begin, as usual, with a few housekeeping items, followed by a brief review of the messages I took away from yesterday's sessions, and then a high-level overview of today's agenda. In terms of housekeeping, I want to make sure that everyone's aware this is a public meeting and it's being webcasted on the HHS website. I wanted to let everyone know that we have sign language interpreters available on the HHS live stream. We appreciate their work to make our broadcast more inclusive. We ask that everyone speak slowly and clearly to assist them in supporting our meeting. For our virtual participants, please remember to mute yourself when not speaking, and please do not use your camera unless you're presenting, asking questions, or answering a question. During discussions, I ask that all members and speakers identify themselves before speaking if I did not acknowledge you by name in giving you the floor. This helps the note taker and others to follow along. Throughout the day, there will be opportunities for committee discussion. If you'd like to ask a question or provide a comment, please send me a message through the chat feature. As always, we will have time for public comment at the end of our session today. This is planned at about 5 p.m. Public comments are not a question and answer session. They represent an opportunity for individuals who'd like to make a statement to do so. The deadline to request a space for verbal public comment during the meeting has passed. However, anyone can submit a written comment up to 3 pages in length to NVAC@HHS.gov. This concludes the housekeeping session. So, for highlights from yesterday, we had a superb series of panels and presentations. We started with a panel speaking to the recent increase in measles in the U.S. and internationally. Inadequate measles vaccination is a major causal factor for outbreaks. My 3 take-home points from this panel were the impact of the hesitancy hangover from COVID vaccination on measles vaccination leaves us in a situation where measles and misinformation together put communities, particularly those with lower vaccination rates, at risk for outbreaks. In my view, this particularly is timely in the context of the current Florida measles outbreak and concerning communications about measles management. The presentation about the recent bio report on the vaccine pipeline and industry investment was notable for the broad number of vaccine candidates in phase 1 to 3 evaluation, but with most vaccine preventable diseases being addressed by only 1 to 2 candidates and only roughly 10% reaching approval, this pool is not very deep. There are also a number of notable monoclonal products in the pipeline, importantly 4 for diseases for which we do not have currently available vaccines. Following a brief break, we had an important panel with experts speaking to aspects of federal, from FDA, ASPR, and BARDA, and CDC, as well as private industry, working collaboratively to create and maintain a strong supply chain for vaccine manufacturing and to minimize the risk of future shortages. Our two late day panels focused on different aspects of childhood immunizations. The first reviewed state policies on immunization for school entry, changes in the environment related to school or state legislation, the impact of a legal decision in Mississippi that opened up religious vaccine exemptions, and options for exemption processes to mitigate the potential adverse impact on school-age vaccination rates. The second discussed the 30-year history and impact of the Vaccines for Children program, as well as recommendations to enhance the program going forward. Finally, we heard an update from the Innovation and Immunization Subcommittee on their report. Work continues with robust support from ANN and OIDP staff and contractors. We will hear more at our June meeting. And we close the day with public comment. We will open today's meeting with expert insights on artificial intelligence and its use in vaccine development and immunization efforts. Then we'll hear from a panel of speakers on innovative approaches to improve adult immunization efforts. After a short break, we'll hear from another panel on vaccinating pregnant women. Our last panel for the day will focus on practices for immunization of special needs individuals. And the day will conclude with our federal agency and liaison member updates and public comment. Finally, as a reminder, please hold our remaining 2024 meeting dates on your calendars. June 13th to 14th, 2024, and September 12th to 13th, 2024. Please refer to the NVAC website for final details on these upcoming meetings. Now, let's get started with our first panel of the day. This panel is entitled Artificial Intelligence, Real Uses in Vaccine Development and Immunization Efforts. With unparalleled scientific growth, emerging diseases, and new technologies, the medical and healthcare sectors have expanded markets, including for vaccine development post-COVID-19 pandemic. One of these newer technologies, artificial intelligence or AI, has already shown to provide huge benefits for public health, but also presents concerns over data credibility. In this session, we'll examine guidance from HHS, as well as explore some broad real-world examples for the use of AI to support vaccine development and other immunization efforts. This work supports the NVAC charge on vaccine innovation, as well as potential new solutions to improve equity by reducing human bias, and lays a strong foundation for advancing precision vaccines. In this session, we'll hear from Greg Singleton from the U.S. Department of Health and Human Services, Dr. Justin Matthew from the U.S. Food and Drug Administration, Ted Schinkelberg from the Human Immunome Project, Mark Langowski from the University of Washington, Dr. Jimmy Golahar from Houston Methodist, and Dimitris Zambas from Pfizer. Mr. Singleton, we have your slides up. And
all right.
I see you on. You have the floor.
All right. Thank you, Dr. Hopkins. First of all, I want to start off introducing myself. I am Greg Singleton, the Chief Artificial Intelligence Officer for Health and Human Services. And of course, thank you to Dr. Hopkins for that introduction and stage setting, and thank Ann and Rebecca for inviting me and helping coordinate with the committee to help present today. I want to talk about a few things today about how we as a department are thinking about AI from just the initial outset, kind of top level. I want to talk about how we are approaching AI as the U.S. government, talk about some principles of trustworthiness that are really important to us and the nation as we deploy artificial intelligence, and then talk about some of our applications, some areas where we are exploring artificial intelligence, and then really hoping to set the stage for the rest of the panel that's going to talk a lot about really the innovative possibilities and the potential for AI in vaccine development. With that, if we can go to the next slide. So here, two simple pictures. And I really ask the question, why artificial intelligence? The first picture we have on the left is data generation. This is a graph of the volume, the quantity of data that we as a species are generating and transmitting and capturing yearly. You see it's a relatively exponential growth pattern there. Right-hand side, we see in this case, it's a graph of the federal workforce as a percentage of the American population. Well, why do I put these two together? The simple answer to why AI is we are generating tremendous volumes of data and information and material on an ever increasing basis. Our population, our workforce, the number of human beings on the globe is not increasing. So our ability to process and manage that data volume, that communication is not really keeping up. So AI comes in as a tool and a capability to help us bring human-like capabilities to this data generation problem. And this problem exists in imagery, it exists in music, it exists in scientific literature, it exists in vaccine literature. The volume is just increasing. So we have the option of either we can take more time to do the work we're doing, we can skip stuff and make an abbreviated job of it, or we can adopt new tools to approach the challenges in front of us. And that's what we're looking at for AIs. How do we use it as a new tool to manage these challenges that we have all across society? Next slide. So when I think about AI and how to frame it, it's important to know AI technologies are not entirely new. The capabilities have increased, and the attention in literature has increased. I think it was the most popular term in headlines, I think, until very recently. Taylor Swift probably bumped that out of the top contenders. But with the development of vast data centers, vast storage, vast communication capabilities, and algorithms, we now have the capabilities to harness these powerful techniques for good. The AI approaches, again, allow us to manage that core challenge, that tension between data generation and, honestly, the limits of human attention. We only have so many human brains that can work on these challenges. That gets to one of my favorite definitions of artificial intelligence, and that is artificial intelligence brings human insights at machine speed. If we can do that, we can accomplish a lot of things and focus on the core challenges for society and science. I'd like to say AI applications are differentiated by how you're using them, the application, not necessarily the technique. So when we look at risk management for artificial intelligence, what's mostly important is what are you doing with it? How are you controlling it? How are you managing it? It's not necessarily that we care so much about whether or not you're using natural language processing or tree-based methods or adversarial models or something like that. It's not the technique, it's the application that we care about. And then as Dr. Hopkins framed out, we are challenged to deal with the practical present of artificial intelligence and the theoretical future. A lot of the news media, popular media, television, TV shows, and movies think about AI in the 10, 15, 20 year out timeframe. What's going to be possible then when we're worried about vastly capable, almost human-like artificial intelligence? A lot of the artificial intelligence we're dealing with now is painting pictures, generating videos, doing genetic simulations, responding to chatbots. In many cases, it's finding the right bit of information, taking it and putting it in the right place at the right time. And that's very different from the AI that we see portrayed in media. So that's what we're trying to deal with as we think about the AI challenges. Next slide. So for the department, we've been working on AI for many years to advance the health sector. But as we've seen, the pace and the capabilities of AI have accelerated a lot recently. So we are taking a renewed look and renewed emphasis on the AI challenges as they present themselves to the department, to the sector. We really hope, we have the opportunity to be a catalyst for successful advances in the adoption of AI, but we need to ensure that we're matching the pace and scale of AI developments. A lot of this comes out in the Artificial Intelligence Executive Order signed at the end of October And through that, we are developing new AI strategy. We're working on implementation roadmap, and really looking at risk management of AI across the department, and then thinking of it from a holistic sector-based approach. Next slide. So I do want to talk a little bit about the Executive Order 14110 on the safe, secure, and trustworthy development and use of artificial intelligence. This is, I think, among the more comprehensive executive orders that I've seen in a long time, where it really works to manage and grapple with the many facets of artificial intelligence, the concerns, the opportunities, and issues that are really important to us as a nation. You can see on the right-hand side here, the sections of the executive order deal with safety and security, citizens' privacy, equity and civil rights, consumers, patients, and student rights, workers and workforce, innovation, competition, and American leadership abroad. And then for the government, our use of AI. And what's important here is, while many of these seem and are risk management approaches, it really is an embodiment of American values and how we want to ensure that we are able to safely and responsibly use these technologies while harnessing the advances for the nation and our citizens. And it's really that balance, that risk management and harnessing opportunity, that's what we're working to accomplish through this executive order. Next slide. I want to turn a little bit to the HHS Trustworthy Artificial Intelligence Playbook and really speak briefly to this. This is a document that we came out with in 2021, and it really precedes a number of the subsequent developments on artificial intelligence. But the goal here and what we sought to do is lay out the principles and concerns and things that we recognize are important for these technologies in order to use them in a responsible manner. So, you see at the top, Trustworthy AI talks about design, development, acquisition, and use of AI in a manner that fosters public trust and confidence while protecting privacy, civil rights, civil liberties, and American values. And that's really what we're trying to do here, ensure that as we're deploying these systems, we're ensuring that they are fair and impartial, that they are transparent and explainable. You understand what they're going to be doing. They're responsible and accountable, and they aren't autonomously operating and doing things that we don't like. We want to ensure they're robust and reliable. They produce what you want them to produce. They respect privacy of individuals and groups, and data is not being used in ways it's not intended. They should be safe and secure, not just from cyber or malicious action, but also safe and secure for people that use these systems. While taken together, the goal is to use these principles to ensure that we have AI that, again, fosters public trust and confidence, so we can feel good with the use of these tools and confident that the risks are managed and we're appropriately deploying these systems for the government and out in the healthcare sector. I do want to highlight that these principles are not mutually exclusive, and it probably is not possible to have 100% on all of these at the same time. And so it's really an important question of risk management, balance, and responsible program design and implementation. Next slide. Turning from the Trustworthy Playbook, I want to speak very briefly about some of the uses that we as a department are leveraging AI for. So talk about some use cases, and many of these are trial, research, experimental systems that we are looking to see if these are useful, that we would consider for further deployment. But we're using AI for things like virtual animal models for toxicology testing. So obviating the need to use, or potentially obviating the need to use real animals for real cell cultures for toxicology testing. Can you use virtual systems to do that? That would be great. Can you use artificial intelligence to identify molecules for drug repurposing candidates for managing pandemics or toxins or weapons or new pathogens that emerge in the environment? Can we use AI to help with tuberculosis detection in chest x-rays when we look at border control concerns? Can we use AI systems for looking at feedback analysis and reviewing public comments because we get vast volumes of information in from the public, and it's important that we appropriately characterize and categorize and understand what the public is saying to us. Can we predict research categories in stem cell research, looking at research applications and documentation? Can we use it to look at text documents and pull out important factors that our analysts or investigators need to consider when they're looking at documents? All these are use cases that we are looking at as a department and hoping to see if there are opportunities to deploy these safely and responsibly. Next slide. And then broadly, we as a department have 163 use cases in the last year, and we continue to develop more, but they really fall in a number of categories around information management, a lot of biology and fundamental research, chatbots, natural language processing, and then detection and device processing through data, images, material, and things that come in to detect features, facets, or important elements. And that's what we see is a lot of these use cases are really on data processing, data management, and we are continuing to work on AI applications in the business operations, business applications, and other areas of just fundamental government work. Next slide. So with that brief overview, again, I want to talk about how we think about AI, talk about how we are approaching as a government, leveraging guidance from the White House, covered a little bit of the trustworthy principles that are very important so that as we use these technologies, that they're trusted and we can have confidence in them. And then talk about some of the applications as a department. As I said, AI applications have the potential to improve care, address health inequities, accelerate innovation, and increase market competition. But we want to ensure we are approaching AI with kind of minimizing approaches that rely on core principles of trustworthiness. It's vital for the nation to both seize the promise and manage the risk to enable progress. Ultimately, we're hopeful that with this careful, measured approach, one day AI will just be another tool in the toolbox that we can use to make progress and improve citizens' lives. With that, I will turn it back over and thank the committee for your attention.
Thank you very much, Mr. Singleton. Our next presenter is Dr. Justin Matthew from FDA. Dr. Matthew, your slides are up. I see you on the line. You have the floor.
Thank you, Bob. Can everyone hear me and see me?
Clear. Yes.
Perfect. I want to thank the panel members for inviting me and giving me the opportunity to speak. My name is Justin Matthew. I'm a policy analyst within the Office of Medical Policy at the Center for Drug Evaluation and Research at the FDA. And today we'll be discussing responsive regulation of artificial intelligence in drug development. Next slide. Before I get started, I want to state that any views expressed in this presentation do not necessarily represent the policies of FDA. Any mentions of specific names and brands are not endorsements. And finally, I don't have any particular disclosures to state. Next slide. So I know Greg touched on this, but I just want to set a baseline. So let's just start with the working definition that FDA currently is using to define AI. And this is derived from the White House executive order published back in October 23. Artificial intelligence is a machine-based system that can make predictions, recommendations, or decisions influencing real or virtual environments. AI systems use machine and human-based inputs to perceive real and virtual environments, abstract such perceptions into models through analysis in an automated manner, and use model inference to formulate options for information or action. Next slide. please. So what are the drivers behind the growth in AI health applications? The main takeaway from this slide is to point to the fact that in the last 15, 20 years, we have gathered a ton of diverse healthcare data that can be linked. With the increase in computing power and breakthroughs in methods of algorithms, all this data can be put to good use to train these models and gain new insights using artificial intelligence. Next slide. And you can see the growth in AI in healthcare research on this slide. The graph was taken from an article published in the journal Nature back in September 2020, a little dated, but still relevant. On the line graph, the authors performed a query search in PubMed using terms machine learning or deep learning and choosing a specific year in the advanced search field. And you can see the exponential growth in the recent years, with 2019 returning search results of over 12,500 articles. And on the right side, it's the same method, but combining all the years. And they broke it down by medical specialty, with pathology, radiology, and surgery accounting for the top three. Next slide. And now this growth is reflected in FDA. Specifically, the Center for Device and Radiological Health, CDRH, has been leading the way. CDRH has authorized nearly 700 AI-enabled devices. And there was a steady uptick in AI-enabled devices starting in 2015 through 2022. More than 3/4 of the devices authorized were in radiology. Next slide, please. AI use in drugs and biologic development landscape occurs throughout all phases, starting from discovery into preclinical research, into clinical research, and eventually in the post-market safety surveillance, and even in manufacturing. Some of the uses in the discovery phase include drug target identification, compound screening, and design. In the preclinical research phase, some of the main applications for AI use are in drug dose range finding, PK/PD type of modeling, and then in the clinical research, AI may be used for site identification, for recruitment in clinical trials, as well as enhanced medication adherence and study retention, which is really applicable for vaccine trials, especially with multi-dose scheduling. Or for example, in the COVID-19 vaccine development, pharma companies use graphical-based knowledge and image analysis to get new insights into illnesses and detect biomarkers 30% faster than human pathologists could. Staying within the clinical research phase, we see applications that are related to use in digital health technologies and clinical trials, and some applications that overlap with real-world data analysis, or even creation of digital twins, where you're simulating placebo response of specific participants. And finally, in the post-market side, AI and machine learning are seen in pharmacovigilance, identifying and evaluating safety signals and adverse event reports. So you can see, again, applications of AI are across the development cycle. But I want to emphasize that some of the use cases may be outside of FDA's oversight, but we still encourage and hope applicants will include that they've used AI in their drug development programs, even if it's outside of FDA oversight. Next slide, please. So within CEDAR, we're starting to see an increase in submissions with AI. This table is from an article published by a colleague, Dr. Chi Lee from FDA, and it was published back in April 2023. You can see in the year 2021, there were a total of 132 submissions that had an AI component compared to only one submission in 2016. With 118 of the 132 submissions being in the clinical research stage phase. So this just reiterates the growth and use of AI in the drug development. Next slide. So all these opportunities will have to be balanced against some of the known challenges with using data-driven technologies. Fundamentally, as with any data-driven technology, it's only as good as the data that goes in. This is especially important for certain contexts of use where we need to understand the data that was used to train these models? Are the data used for AI development of high quality? Are there any inherent biases within the data? Is the data representative of the intended population? With regards to opacity, you may sometimes run into the black box paradox of AI algorithms and trying to understand how the model produced a particular output. I also want to touch on the necessity for transparency, and not towards us regulators, but also the end users, the providers, the patients for whom the algorithm have direct impacts. And now having all this data on individuals now opens up data privacy concerns, and having robust security infrastructures are key and important. A particular challenge for FDA is faced with is its oversight and governance of AI as it pertains to adaptive algorithms after approval and once deployed to the public. Next slide, please. As I mentioned previously, my colleagues at CDRH have been leading the way with regards to having experience in submissions containing AI. They have had several workshops and published some guidances for use of AI in medical devices. And I want to highlight that in fall of 2021, FDA's CDRH, in collaboration with Health Canada and the United Kingdom's Medicines and Healthcare Products Regulatory Agency, jointly identified 10 guiding principles that can inform the development of good machine learning practices for medical devices. Next slide, please. I'm not going to read all 10 guiding principles, but I just want to touch on some key aspects. The first principle I want to highlight is the incorporation of a multidisciplinary approach from the onset of model development. It's not just important for data scientists and biostatisticians and epidemiologists to be involved in the initial phase of the model development, but we should be including clinicians, pharmacologists, other impacted parties, and thinking around developing these models so that you're developing the models on robust data from the onset. And what I mean by robust data is it well representative of the intended patient population? Are there any hidden biases? Because the algorithm developed on the training data set may produce excellent results initially, but once deployed to the wider intended patient population, the algorithm may not work as well. And finally, maintaining a human in the loop approach during validation is important, especially when AI model has high level of influence and the decision consequence is high as well. Next slide, please. Now, I want to emphasize engagement is key here. These are new frontiers, and as we work on navigating through the processes of how best to implement and regulate AI in the development of drugs and biologics, as well as within the life cycle of medical products, we want to promote a mutual learning on three main core principles, which are 1, human-led governance with accountability and transparency, 2, high quality, reliable, well-representative data, and 3, the AI models themselves, from development to performance testing to monitoring and validation. So what's next? The White House executive order establishes a government-wide effort to guide responsible AI development and deployment through federal agency leadership, regulation of industry, and engagement with international partners. CEDAR specifically is especially tasked with developing a strategy for use of AI and AI-enabled tools in drug development. So we in CEDAR, in collaboration with the Center for Biologic Evaluation and Research, CBER, with help from CDRH, are developing a guidance for use of artificial intelligence to support regulatory decision-making for drugs and biologic products. With regard to advancing safety and security, we see utility in the use of AI in pharmacovigilance and post-market safety, and FDA will continue to use grants and other funding for demonstration projects. And just like everybody else, the FDA is looking to use AI to help us internally increase productivity and efficiency as well. And like I mentioned in the previous slides, we want to promote industry engagement. That is why we hold public workshops. And in fact, CEDAR will be organizing a public workshop with regards to the use of AI in the drug development cycle in the summer. So look for further details to come out soon. And on that note, I will pass it back to Bob.
Thank you very much for your presentation, Dr. Matthew. Our next presenter is Ted Schinkelberg from the Human Immunome Project. I see you on the camera and your slides are up. You have the floor, sir.
Great, thank you. And great presentation, Justin. Super interesting here what the FDA is doing. My name is Ted Schinkelberg. I'm actually managing partner at Next Frontier Advisors. We're a network of consultants that help NGOs, companies, and philanthropy with vaccine development from design through manufacturing, and former co-founder of the Human Immunome Project, a global NGO focused on understanding the immune system. Next slide. We're going to move pretty quickly here through some technical material, but I want to give you a sense of what's happening in biomedicine and biomedical research and the application of AI, advanced computing, machine learning. And in biomedical discovery, we're really starting to see changes and impact driven by AI. One example is computer vision, which was really optimized on the internet. Retinal scans are helping identifying underlying disease just from the image of the retina and with high degree of accuracy. So the retina scan is able to predict kidney disease, Alzheimer, cardiovascular diseases. And again, I'm just going to go through a couple examples where AI has had an impact or is having an impact on biomedical research. In the protein design and prediction area, Google's spin-out DeepMind developed a program or algorithm called AlphaFold, which is able to predict structure of protein structures from amino acid sequences. And they do this now for 200 million known proteins with a reasonable degree of accuracy. Previously, it would have taken up to a decade sometimes to develop the structure of a single protein. So this is really accelerating how we're understanding core components of our biology. And the last thing I'll just highlight here to give you context of generally how AI is impacting biomedical research. Recently at MIT, a deep learning algorithm identified the first new class of antibiotics in a generation, and it did so relatively rapidly, completely different context than sort of the laboratory-based chemical process that we've been using for antibiotic discovery. So that's a brief snapshot. There's other areas we could obviously go into, but I want to keep moving. Next slide, please. In the history of vaccine development, technological advances have driven our advances in our ability to design vaccines. And this goes back to the 18th century through the 21st century. And this is a slide that's adapted from Stan Plotkin's vaccine book. And even in the first couple decades of our own century, structural biology, mRNA, and synthetic biology have yielded vaccine for RSV, COVID-19, with new platforms and new ways to design adjuvants, which give us greater potential on protecting individuals. So technological advances often are the drivers of ability to design new vaccines. Next slide. I'm not going to dwell on this, but there's a lot of investment, a lot of hype, and a lot of activity that's going on in the AI biomed space. This slide's probably already outdated, but this gives you a sense of what's going on now, and kind of the hope of AI within biomedicine on the product development side. Next slide, please. We're going to turn now to vaccine development, and we're going to use this very broadly to think about how AI and approaches in advanced computing can help solve some of the problems or hurdles that are hindering effective vaccine development. And I'll give you sort of quickly some examples in each of these areas, but they relate to our ability to better understand human immunity and have interventions that are able to protect key populations, our ability to design vaccines better, and our ability to optimize how the vaccines are developed as well as tested. So a number of these areas include kind of modeling and prediction of both the systems, prediction within and modeling within key populations, understanding how to design or identify key antigens or receptors where it's really where the rubber hits the road of immune protection, and optimization. So we'll go through each of these. Next slide, please. And I want to just, offer a little bit of temperance here, hype and reality. AI and advanced computing offers tremendous potential. We saw that on the first slide, but this is far from realized. And most of these technologies are still nascent, particularly in the vaccine area. And they need to really demonstrate impact, efficiency, effectiveness, and efficacy. And we're not there yet, but it's starting to change things. And I just have a brief quote from Jim Crow, who runs the Vanderbilt Vaccine Center, which basically says a lot of the current laboratory approaches are still more efficient and effective than a lot of the approaches in AI. That may not be true in the coming years, but it's true right now. And AI really needs to demonstrate, and scientists need to demonstrate the effectiveness in real world settings of ability to protect people and extend lives. Next slide, please. So what's driving all this? And we saw in previous slides from the other speakers that, data, the ubiquity of data, and which is really the fuel for AI. And on the biomedical side, we're seeing new types and depths of data that we've never seen before. And a lot of this data can be generated at lower and lower cost, particularly in the genomic setting. From small drops of blood in systems biology, we're basically able to look at almost every component in the body, from our genome, transcriptome, proteome, microbiome, et cetera. We can generate a lot of data about how biological systems are working. And with that, we are able to take advantage of some of these accelerated advances in computing. If we combine that within population or clinical studies, longitudinal studies, that looks at populations over time, taking samples, we really are able to get a dynamic look at individuals and how their biology changes either by infection or vaccination, and understand drivers of better protection, better vaccination versus non-response. And we're having an increasing amount of structural and molecular data, which is going to help us design vaccines. Next slides, please. So let's look at a couple problems that are underlying our ability to develop both therapeutic as well as prophylactic vaccines. I'm also going to throw in immunotherapies because we kind of look at the immune system as one system that helps us fight disease. We don't really understand how the immune system works. We don't really understand the drivers of effective vaccination other than maybe antibody titers and some levels below that. But this is a complex distributed system that has memory and changes over time. And if we were able to understand at a systemic or component level how to model or create predictive structures that tell us how the immune system works, it would really accelerate our ability to design and vaccines or other interventions to fight disease. Next slide. So I'm just going to highlight, without going into a lot of details, some advances that are occurring within the field of immunology underlying future vaccine or future immunological interventions to protect us from disease. This is a study out of the Sanger Institute in the UK. And basically, they developed a proof of concept mathematical model, which looks at the immune system as a distributed system, and it predicts interactions from protein molecules to multicellular behavior across organs. And this is a breakthrough study in terms of understanding the immune system as a system. It's the first step, it's early, but it's super interesting to see what mathematics and modeling can do. Next slide. This is a study that we did at the Human Immunome Project where we took systems biology and small drops of blood to understand and parse out who responds to vaccination and who doesn't. And this was to the HBV vaccine. And looking at early immune signatures in the innate immune system, we were able to predict who was going to respond and who wasn't prior to vaccination based on immune signatures, as well as a level of antibody response. And so this was a very small cohort, but it starts to show the power of machine learning and AI in terms of predicting a system and really parsing out the immunological drivers of protection, which we don't yet understand for vaccination. Next slide, please. Another major problem in our ability to protect individuals is the variation of immune responses across populations. Our immune systems are really different at the beginning of life, pregnancy, infants, to end of life, older adults. Those are immunocompromised, and those who are living in environments that are highly stressed, like those in the low and middle income countries. These groups make up the largest burden of disease in the world, and yet we're not so good at generating effective immune response or vaccine responses in these groups. Next slide, please. This is a study done out of Stanford, which takes a systems biology approach with a longitudinal study, and it starts to look at immunosenescence and the drivers of our declining immunity as we age. And it developed predictive signatures from blood samples and analysis of gene expression, cytokines, cellular phenotyping of immune age. And this was highly correlated to mortality, even more so than certain risk factors in the Framingham Heart Study. This is getting at the drivers of the decline in our immune system. And if we can understand that, we might be able to engineer better vaccines to protect an aging world. Next slide, please. Another problem, major problem, particularly for complex infectious diseases as well as non-communicable diseases, is the identification of antigens and their immune receptors, which are protective against disease. This is where the rubber hits the road in vaccination, either therapeutic or prophylactic. This is where we develop antibody or effective T cell responses. We don't really know how to model this. Next slide, please. This is a super interesting study in cancer immunotherapy, which used a deep learning algorithm to identify specific T cell receptors that were associated with response for immunotherapy. So which T cell receptors protected people from or helped clear cancer cells in their body. This is a general concept that could be applied to many different vaccines and could help us design interventions to not only have solid antibody responses to vaccines, but effective T cell responses to vaccines if we can understand the specific receptors or markers which are associated with response to vaccination or immunotherapy. So it's a very interesting, powerful study, at least in concept, using deep learning. Next slide, please. This is a study which used a general language model to help accelerate the natural antibody evolution or affinity maturation. This is the process by which an antibody is evolved in the body to bind more and more, better and better to antigens, and basically help more effectively neutralize infections. And so this was a study in which in 2 rounds of computational evolution, they were able to evolve better antibody effectiveness and binding for coronavirus, Ebola, and influenza. Super interesting study because this is hard to do in the lab. And this gave a computational approach for modifying antibodies and underlies our ability to design maybe future vaccines or monoclonal antibodies through a general language model approach for antibody evolution. Next slide, please. So another problem is a lot of our vaccine development and platforms aren't optimized, and this hinders our ability to have effective vaccines, to distribute vaccines, and our ability to respond to pandemics, and particularly mRNA, which is a very powerful tool for responding to pandemics rapidly, has a number of issues around stability, manufacturing, cost. And so next slide, please. One of the questions that was asked is, can we improve stability of protein expression for mRNA by applying a computational approach which was used in linguistics? And surprisingly, this algorithm suggested new ways to tweak mRNA molecules for COVID-19 vaccines and design vaccines which have better chemical stability, protein translation, and immunogenicity. Again, this was done computationally, and these are kind of in-lab markers, but it pointed to a way to rapidly design and optimize this platform better. Next slide, please. So another question, which I think was, teased out slightly, a little bit in the previous presentation, is can we optimize clinical studies? Currently, for vaccines, clinical trials are hugely expensive, time-consuming, and not really that predictive in the earlier stages. So can we combine learnings from systems biology, where we're understanding the drivers of immunization or safety much better, particularly in key subgroups, with algorithms that help us parse out the selection of participants? Next slide, please. And so in the future, you could start to see efficacy trials that instead of being in the tens or hundreds of thousands of individuals, in the hundreds or thousands, which are more stratified, driven by biology and driven by the identification or knowledge of predictive signatures. This would give faster results, greater probability of success, and include a lot of different biological markers that are potentially associated with protection of populations or safety. Next slide. And way off in the future, maybe, could we move towards AI simulated vaccine trials? This may or may not be on the regulatory path, but it certainly would help us to have a better idea of bringing a product, a better idea of de-risking a product before it goes in the clinic. Could we run a simulated trials based on what we know more about human immunology, using supercomputing, using AI, to sort of say this is a good target or this isn't a good target, and have predictive outcomes through complex models of the immune system or populations about whether vaccines will or won't work. This is off in the future, but you could see this really changing the process of vaccine development and clinical studies. Next slide, please. This is the last slide I just want to summarize. New technology has always been a driver of our advances in vaccines. AI probably won't be any different. It's already changing many other industries, from investment banking, security, imaging, media, but we're really, really early on. We're likely to see new tools like large language models, which are really good at complex, noisy data systems that are annotated. But the proof is in the pudding. We got to show clinical efficacy. We got to show improvement in efficiency over current clinical or lab approaches. And we have to start showing these really interesting concepts that are in the lab to see if they actually work in protecting people, extending people's lives, and improving our ability to combat diseases. Next slide, please. And I'd just like to acknowledge a number of institutions and individuals who helped contribute ideas to this presentation. Thank you.
Thank you very much, Mr. Schenkelberg. Our next presenter is Mark Longowski from the University of Washington. Mark, your slides are up. See your face. You look like you're still muted. You have.
Yeah. Can you hear me?
Yes, you're loud and clear. Go.
Awesome. Thank you. Hi, I'm Mark. I'm a senior graduate student in Neil King's lab here at the IPD. Neil couldn't be here today because he had a conflict that came up, but I'm going to talk a little bit about some of the AI-assisted vaccine design that we're doing here in the lab. Next slide, please. So previously, people here at the IPD have developed these self-assembling nanoparticles using computational methods, using some of these biophysical-based models. So this example right here is in the gray and in the purple are two existing proteins that you can dock together. And then you can, in some sort of symmetric arrangement, in this case, it's an icosahedron. And then you can design interfaces, de novo interfaces between them so that they will, when mixed together, form this nanoparticle every single time. And next slide, please. And so what you can do with this is, so this same nanoparticle that was designed was used as a scaffold to display the SARS-CoV-2 RBD, which you can see on the left. And it was genetically fused, and this was done in collaboration with David Wiesler's lab here. And in the center here, this vaccine is called SkyCovione. In the center here, it has a higher neutralizing titer than AstraZeneca's SARS-CoV-2 vaccine. And so, this is proof here that this is the first, again, computationally designed protein medicine, and that this can work, right? And this was, I think, approved about a year and a half ago in Korea. Next slide, please. But now, okay, so these are the old methods, but what can we do now, right, with AI-assisted protein design? And so we kind of think of this as a pipeline. So on the left here, we have protein backbone generation, right? So what features do we want, right? So like we need to make the skeleton of the protein. And then the next step is sequence design. Okay, so how do we make a sequence that folds it into this shape every single time? And then the actual structure prediction networks, right, like AlphaFold, to say, okay, feed it that sequence and see does it actually fold into the design model? And the last step of all this is, after we've designed all this in the computer, to do experimentation with it, right? And see, determine structures in real life and see if they actually match the design models that we made in the computer. Next slide, please. And so David Baker, the head of our institute, has been at the forefront of this. And so he used a lot of machine learning to for each of these steps, right? So in the first box here, hallucination, inpainting, and RF diffusion are ways to generate these backbones using these AI assisted networks. In the second box in the center, protein MPNN is another machine learning algorithm that designs sequences for these backbones. And then third, as has been mentioned before, but AlphaFold and what was developed here is RosettaFold to actually predict from sequence highly accurate or have highly accurate predictions of structures. Next slide, please. So as an example here, protein MPNN. So it can take in a backbone. So the way protein MPNN has been trained is that it's been trained on the PDB, so from all existing structures. And basically it's learned features about what makes like a good sequence. And so what you can do here on the left, or for this, this alpha helix in pink, is that you just get a backbone. So maybe you generate something from the methods that I mentioned previously, and then you can feed it through the protein MPNN network. And then protein MPNN is going to give you a sequence that it thinks is really good for that specific backbone. And then you can take that sequence and predict it and see how it works. And then next slide, please. So when you used protein MPNN to apply sequences to newly designed backbones, so on the left here, what we're showing is soluble yield. So MPNN designed proteins have a much higher soluble yield compared to AlphaFold hallucinated proteins. And on the right is CD spectroscopy data. And this is basically just looking at the secondary structure content of these proteins, but at 25C or 95C, the spectra is nearly equivalent, right? So these proteins are really, really stable across a huge or a large temperature range. Next slide, please. And so going back to the backbone generation, so at the cutting edge is RF diffusion, right? And so at the bottom here, this method's been inspired by deep learning methods to make synthetic images like DALL-E, maybe some of you have heard about. And so the way those models work is that you train it on a bunch of images, and basically you noise those images. So you can imagine that picture going in reverse. You make it more staticky, more noisy, and then you're training the model to be able to progressively, step by step, eventually generate something into, that looks like an actual image. So what you're seeing at the bottom here is actually on the right, that picture is of somebody that doesn't exist. This was generated by DALL-E, right? And again, this is just from what it's learned about images. And we can do the same thing for proteins. So at the top here in the upper left, you can see that you have this like noisy little cloud of atoms that eventually form into this backbone that looks like a protein, right? And so we've trained, or people here in the Baker Lab have trained these networks on the PDB, so on existing structures. You noise those structures, you teach the model how to like re-noise it or denoise it back into something that looks like a protein, and then we can use these to generate actual protein backbones. Next slide, please. And so RF diffusion can accommodate, a bunch of different tasks. So you can make just proteins unconditionally, as shown in the upper left. You start with a cloud of atoms, and then it forms into, denoises into something that looks like a protein. On the right, you can make a protein binder towards something. So say you have some sort of receptor you want to target. In green there, you can choose a spot on the protein and say, okay, diffuse a backbone that will fit into that spot on the target protein. In the lower left, you can make symmetrical oligomers. So this could be like a nanoparticle, or it could just be like a dimeric or trimeric protein, whatever you want. And you can diffuse symmetrical oligomers that way. And in the lower right, at least in the context of maybe like vaccine design, you could do functional motif scaffolding. So you could take an epitope, right? A known epitope that maybe you want to immunofocus things towards. You can pull that out and actually tell diffusion to, build something completely new that'll support it. And next slide, please. And this is one example. Okay. And so going back to kind of like the very first slide, is that before we had to use existing proteins, dock them together to make a nanoparticle, which maybe isn't necessarily the most custom way to do things, but obviously it can work. But here now we can actually diffuse these nanoparticles completely de novo. And so Helen in the lab made this particle. So she diffused this icosahedron shown on the left here. So it has 60 copies of a protein. So basically we told diffusion to diffuse something that looks like an icosahedron, right? Generates this backbone on the left, use protein MPNN to design interfaces between everything. And then on the right is an actual cryo-EM micrograph. And those are the averages that are shown there. And below it is the cryo-EM density. And so when you fit the design model into the density, you can see that it matches really, really well, right? And so this is kind of the new way that we're making nanoparticles now, and it's much easier compared to, or in theory, compared to the old methods that I showed at the beginning of the presentation. Next slide, please. And so an actual example for an antigen, in this case, this is HIV envelope trimer, is that, so there are two parts to it. There's GP120 and GP41. And so there's been a lot of work done in the past couple decades to stabilize these HIV envelope trimers. And so on the left here, it's showing that there's a metal stable core. It's not fully resolved. And a lot of the stabilization mutations that have been applied to this protein to make it, make it more happy and actually keep it as a trimer for vaccines are towards this, the part that's called GP41, which is like the trimerization interface. And so the thought here is that kind of like we hit our limit in putting Band-Aids on it to try to fix it and make it stable. So the question was, okay, how can we use these new models that we've come up with in the last few years to make a completely new core and take away the problem, right? So basically the outside of the HIV envelope trimer is going to look exactly the same, but the inside now is going to be a completely de novo interface. And so on the right, the thought here is that, okay, can we remove the GP120 core? Maybe we'll have to do some stuff to play with the GP120 permutation and provide a new supporting structure. And one advantage of this is that there are some germline targeting vaccines for HIV that only show a monomeric or just one copy. They don't actually show the full symmetric representation of this trimer. And that potentially could lead to off-target responses. So the hope here is that you make something that's higher fidelity that can elicit the vaccine response that you actually want. Next slide, please. And so this is work all done by a visiting postdoc, named Johan Aldon, who's at Amsterdam UMC. And so he used some of these AI-assisted methods to generate these new cores for the GP120 trimer, and also directly fuse it into an existing nanoparticle. And so at the top here is showing this is a 2-component nanoparticle called I53DN5. In blue is the trimeric component, which we want to scaffold the trimer on. So in the lower left, I53DN5B, one of these components was used as the base. We used oligomeric hallucination, one of these networks, to generate a helix bundle. So you extend it out. And then the next step to this now is you dock the GP120 trimer and find like the best orientation. And then you in-paint, so it's another model, another deep learning model, to actually connect the GP120 trimer to this helix bundle. And then you use protein NPNN to design everything to make sure that the inside of the GP120 and this newly designed de novo protein bundle are, actually make a proper interface. And then next slide, please. And so this actually works. It's a pretty incredible result, but this is cryo-EM structure of this GP41 free native-like trimer. So on the left here is the design model. And so what's highlighted in pink there is what the de novo backbone that's been generated at the core of the protein. And then was designed with protein NPNN. And to the right of it is this GP41 free trimer overlaid on a SOSIP trimer, so like a native envelope trimer. And this is in complex with VRC01 class antibodies. And you can see that there's almost no difference between these two things. They're pretty much identical to each other. And then on the lower left is the actual cryo-EM data. And you can see that little helix bundle at the bottom that was diffused out. And then the antibodies bound to the trimer. And then to the right of that this is just showing 3 trimers on the nanoparticle. That's what's sitting below it, the soccer ball below it. You can see that there's antibodies bound to it and that, it looks like it's in the proper orientation as it was intended to be designed. And so when you have a fully occupied particle, you have the 20 copies of envelope trimer and you have 60 Fabs binding each binding site that is available. And so this is going into, currently it's going into mice to be tested. And we have some other examples, which so I work on malaria, which I couldn't show today, but we are testing these things out for stuff that's been generated with all these, deep learning protein design methods. And next slide, please. And so just to summarize, right, so these computationally designed protein vaccines are a reality, right? So even with the old methods, right, you can make these protein vaccines. And AI has changed a lot of how we've designed proteins. Before at the start of my PhD 5 years ago, what we started with biophysical models. And now, we still use some of that, but these deep learning methods are way faster and way better. It's not perfect by any means, right? And there isn't proof yet that they'll, necessarily work, or a guarantee that they'll work. But it's been amazing what has been done in the last few years. And then the last line is ambiguous and a bit vague, right? But I think, there are concerns. I'm sure people have seen the news, right, about AI. But at least in the context of protein design, we think it's a really positive force in designing these better medicines, right, and vaccines. And there's a bunch of work coming out of the Baker Lab that you can see, whether for biologics and therapies like that seem to be really, really promising. And again, same thing in our lab for the production of actual vaccines. And next slide, just to acknowledge the people that have worked on all this stuff, the data I presented here. Neil, of course, my PI, Helen, and Yoan. And thanks to all our funders. And thank you so much.
Thank you very much, Mark. Very good presentation. Our next presenter is Dr. Jimmy Gallagher from Houston Methodist. Dr. Gallagher, your slides are up. I see your face. You have the floor.
Okay, great. Thanks. First of all, thanks for having me. It's an honor to speak with you all and share how we're beginning to use artificial intelligence and machine learning to design and validate immunogens to different viruses. I'm also excited to share a little bit about how we safely generate data in the laboratory to enhance these models and make them better over time. Next slide, please. So I thought we should start by taking a look at traditional vaccine design, which sets the stage for appreciating what AI can do. So first of all, traditional vaccine development follows a sequential and structural workflow, which we dissect into 4 key stages. In the first stage, this is antigen design, where we rely on rational design, directed evolution, and computational methods, mostly physics or evolution-based. These methods then attempt to craft antigens that resemble some conformation of a protein, usually pre-fusion for viruses, and then hopefully elicit a desired immune response. Moving to the second stage, we must test these in vitro experiments to assess the various properties of the vaccine candidates, such as stability, epitope presentation, expression levels, antibody binding, and even structural integrity, like the cryo-EM images you just saw. These experiments are vital for down-selecting designs. The third stage involves preclinical work, where we're working in small animal models to understand toxicity, and then moving into larger, more physiologically similar animals to challenge the vaccine's efficacy and further assess safety profiles. And then finally, the best candidates might make it to the clinical stage, where we're going into humans to assess efficacy and safety as well. Throughout all of these stages, the traditional approach has always been about abstracting concepts from the data and optimizing those candidates through trial and error. And as you can see with the arrows, it goes in both directions. So as we move from stage to stage, we learn more, which enables us to design or engineer better immunogens. And as you can imagine, this is an incredibly time-consuming effort that often takes years, decades to be successful. As we'll see in the next slides, we believe that machine learning has the potential to revolutionize this traditional workflow, making it faster and more efficient. Next slide, please. So moving beyond the traditional vaccine engineering, our goal is to build a platform that integrates these specialized tools into a comprehensive framework for immunogen design. We build purpose-built tools that are trained to abstract very specific types of information and in turn design or constrain vaccine candidates based on that information. So first, we start with antigen identification and pre-processing, which is currently our in-house genomic surveillance efforts and even links to public databases such as VIPR, run by BVBRC. Here we determine the type of pathogen, identify potential antigens to use as immunogens, and then prepare them for our other modules. Once we have antigens identified, our AI-driven tools then analyze sequence and phylogeny to understand the evolutionary relationships and sequence variability of pathogens. The structure and stability tools use modules to model the 3-dimensional structures and then predict their stability and enhance the presentation of protective epitopes to the human immune system. And then, of course, immunological profiling is another critical component where we're expanding the tools to understand and predict how human immune systems might interact with different antigens. All of these purpose-built tools feed into our antigen design model, where we are testing different multitasking architectures to synthesize data from the various modules to propose the most promising vaccine candidates. Now, I'm going to go through each one of these and provide a single example of a tool for each of these modules. So next slide, please. Okay, so we're starting with sequence and phylogeny. Here we're developing models for very specific viral families to inform design. This particular module is broken into 3 phases: training, generation, and folding. In the training phase, we use a large language model, which you've heard about, adapted for proteins to learn the sequence distribution of a viral family. This training involves understanding the complex language of proteins. So those are the amino acid sequences that determine their structure and function. The generation phase is where the trained model becomes an architect. It auto-generates a diverse set of new protein sequences adhering to these learned patterns. These sequences are not just random guesses. These are hypothesized to maintain a biological structure and function that we want. We further refine these processes through techniques like mean pooling and k-means clustering to select sequences that represent our desired traits. And lastly, we have the folding phase. So once we've generated a new sequence, we want to put it into a 3-dimensional structure that we can look at. And you just heard about AlphaFold, Rosetta Fold. We use these tools, OmegaFold, OpenFold, and this allows us to predict the structures of our generated sequences. And this is a really important step for proteins that don't have structures. And we can also take this output and pipe it directly into our structure and stability tools that I'll talk about a little bit later. Next slide, please. So here is an example of using sequence and phylogeny. This is an example, a subset of paramyxoviruses. To the left, we're looking at genetic diversity within the Henipavirus branch. The genetic relationships between these viruses are shown by the color coding. The highlights are the geographical spread of the Henipavirus genomes. On the right, we dive into the structural biology, where we're examining how far the stabilizing mutations, such as the disulfide, make it along the evolutionary tree. So understanding the sequence and phylogeny of a particular virus enables us to make faster predictions, and in some ways is automating the process that was used for the COVID-19 vaccines. As you probably know, the McClellan laboratory had previously stabilized the MERS spike protein. I think they published that back in 2016. And then when the Wuhan virus sequence was released, they were able to solve the prefusion stabilized structure of that within about 12 days. So these prefusion stabilized mutations are also found in all US approved vaccines. So structure and function and sequence and phylogeny are all very closely intertwined. Next slide, please. Next up is our structure and stability algorithms, where we use computer vision to engineer proteins with desired conformational stability. For most cases, we're using these algorithms to stabilize the prefusion conformation of a viral protein to elicit protective monoclonal antibodies. In this module, we're teaching our neural networks very specific chemistries that immunogen designers like Jason McClellan or Andrew Ward or Erica Sapphire or Peter Kwan would use to rationally design variants. So we're teaching the net how to look at a protein structure and make decisions on how to best mutate the protein to lock it into a particular conformation. This includes teaching the net where to put proline caps, disulfide bonds, cavity filling substitutions, locking key mutations, salt bridges, and even indels. Next slide, please. So this slide illustrates our cavity filling mutation module. The process begins with scanning the protein to calculate solvent accessibility, identifying residues that are buried within the protein structure. These residues, depicted here with varying degrees of solvent accessibility, are critical targets for stabilization mutations. Once the targets are identified, our net is designed to recognize and down select specific amino acid substitutions. In this case, isoleucine, leucine, methionine, phenylalanine, which are all known to influence protein core packing and stability. The net effect of these mutations is then analyzed through another program that we've developed to build variant structures and calculate changes in cavity volume. So as you see in the transition between valine to leucine, our goal is to ensure that these mutations lead to a reduction in cavity size, which will then correlate with increased structural integrity and therefore potentially greater stability of vaccines. By meticulously optimizing these structural parameters, we're able to design proteins that not only meet our stability criteria, but are also more likely to retain their shape and function when introduced as immunogens. Next slide, please. Another critical component of our immunogen design process is immune repertoire profiling. So it's very important to understand how the immune system responds to immunogens as well as natural infections. And so on the left side of your screen, I'm showing you B cells. These are where your neutralizing antibodies come from. And we want to catalog the types of antibodies and epitopes they recognize for natural viruses and immunogens that we make mimicking those viruses. We're looking for protective epitopes, and we want to avoid and even mask non-protective immunodominant epitopes in our vaccine designs. We also care a lot about how T cells are elicited by immunogens, and so we want to study the natural response and protection afforded by cellular immunity. Next slide, please. So this slide is a little bit deeper dive into T-cell immunology and tools that we're developing. This is a joint project with the J. Craig Venter Institute as well as the La Jolla Institute for Immunology. So thanks to Jean Tan, Alba Grafoni, and Alex Setti. Here we're focusing on the identification of conserved immunodominant T-cell targets. The crucial task here is selecting taxonomic groups and determining the conserved regions of these viruses, right? And those become priority targets as indicated by the sequence alignment on the left. In the central part of the slide, we're outlining the methodology. So we perform a meta-analysis of known T-cell epitopes using the Immune Epitope Database, an analysis resource that's been curated by Dr. Setti for many, many years now. This feeds into a machine learning algorithm where we look at conserved regions of the antigen and design new immunogens aiming to elicit cross-protective immune responses against viruses that are closely related. Step 2 is the integration phase. So this is where we bring together results from epitope analysis and predictions. This allows us to select a set of candidate epitopes that we can use for experimental evaluations and then maybe use in our immunogens. This pipeline allows us to prioritize epitopes with immunogenicity and high conservation, as these are likely to produce the strongest immune responses. However, we're also looking at those with moderate conservations that they exhibit high immunogenicity. This ensures a robust and comprehensive immune defense. Next slide. So we're also teaching our neural networks how to target specific immune populations with mRNA. So as I'm sure you're aware, mRNA is in a lipid nanoparticle that gets into the cell, is translated into a protein, and then either makes it to the surface in the case of B cell presentation, or gets processed by cellular machinery in the case of CD8 presentation that would go through the proteasome and MHC1 molecules. We also have pathways to hit CD4s, which would go through the lysosome. And we believe this strategy will create a robust immune response by activating both arms of adaptive immunity. By targeting these very specific pathways, we can stimulate a more comprehensive and perhaps more durable immune response. Next slide, please. Right. So from silicon to carbon. So computational predictions are great, but they really don't mean anything until they're tested in the real world. To putting them in carbon is what we say in our laboratory. For the B cell targeted antigens, we employ mammalian display, which is a technique that we developed during the COVID-19 pandemic to understand how mutations in the spike protein were impacting therapeutic monoclonal antibodies. This platform enables the rapid characterization of surface-expressed viral glycoproteins. We use the platform to understand expression, stability, antibody binding, and even host receptor interactions. We also designed the system where we could cut the glycoproteins off the surface and structurally characterize them with negative stain EM. And of course, this first generation could be done in hundreds of variants of glycoproteins in a matter of days after we had sequence verified DNA. The second generation of the platform saw improvements in scale, where we built stable cell lines. This allowed us to perform library-based approaches like deep mutational scanning and mammalian cell lines. And that's where we can put all 20 amino acids at each position of a protein and determine what those mutational effects have on stability, antibody binding, or other properties that we may be interested in. We also increased the number of variants that we could test by several orders of magnitude by moving to this library-based approach. It was also at this point where we realized that the platform could be used for engineering and show that we could replicate pre-fusion stability across coronaviruses, which is shown at the bottom. So this detailed workflow really epitomizes the transition from theoretical design generated by AI algorithms to tangible experiments in mammalian synthetic biology that allow us to validate and refine these designs. We can also use this platform to train models for very specific tasks, expression, stability, antigenicity, and this really creates a feedback to improve our prediction algorithms. Next slide, please. So moving beyond coronaviruses, we've also begun using this platform for many other viral families. Here I'm showing some arenavirus glycoprotein data, and using our mammalian synthetic biology platform, we also use conformational antibodies as probes to report on the structure on the surface. So it's unrealistic to test the conformation of all your AI designed proteins, but using conformational antibodies as probes allows us to test a million designs in parallel. In this example, we're showing lots of fevervirus glycoprotein binding to 2 known neutralizing antibodies. These glycoprotein variants were predicted and validated within a couple of months. And you're looking at the combination of AI-validated mutations. The original best in class is shown in pink here. So in just a couple of months, we were able to increase binding to known neutralizing antibodies that took about 10 years to originally produce. The spheres in the blue represent GPC variants that warranted further characterization, and I'll show that on the next slide. So this table shows the fold change in binding affinity of the various AI-generated immunogens relative to the previously engineered version. Each row represents a unique antibody that binds to a particular epitope class. The heat map indicate the fold change in binding. The darker blue, the better. This allows for a rapid visual assessment. And as you can see, by looking at all of the known, or as many of the known antibodies that bind the loss of GPC, we can start to see where synergy happens from these mutations, and where also where we're breaking interactions, and those are shown with less binding in the red. This forms the basis for selecting candidates with enhanced antigenicity profiles for further development, streamlining the process of immunogen optimization and allowing for more targeted and effective vaccine design. These data, again, can also be used to strengthen our models for future predictions. And so on the next slide, I'd like to thank my CEPI-funded collaborators. This truly takes a village. Dr. Jason McClellan is our structural virologist. Dr. Scott Weaver and Alexander Freiberg from UTMB Galveston handle all of our BSL-4 work. T-cell immunologist Alessandro Setti and Alba Grifoni from La Jolla Institute of Immunology helps out quite a bit. Jean Tan, a virologist from the J. Craig Ventnor Institute, as well as Jim Davis and Arvind Ramanathan from UChicago or Argonne National Labs with help with bioinformatics and some of the large language model that you've seen. Next slide. And finally, I'd like to thank the leaders of the team leaders of my group, Antibody Discovery and Accelerated Protein Therapeutics, for all of their help. Everything that you have seen is due to all of their hard work and leadership in our lab. And I will stop there. Thank you.
Thank you very much, Dr. Kalahar. And our final presenter in this panel is Dimitris Zambas from Pfizer. Your slides are up. I see your face. You have the floor.
Thank you very much. I'd like to start by extending a big thank you to the organizing team, NVAC and HHS, for inviting me to present this use case. It's a very different type of use case from the last three that we heard. I think the whole industry appreciates in those very scientific, detailed use cases. You obviously need that detail in defining your approach. In the operational space, we have fallen victim over a number of years in designing our use cases to be very broad, in a sense, looking for some magic bullet that would somehow accelerate the clinical development process. This use case is very specific, and I think it's one of the reasons that it was successful is because we approached it not looking at the entire study development or clinical development continuum, but looking at very specific components within that continuum that could have a meaningful impact in the overall timelines. Go to the next slide. One of the heavy lift, if you will, of executing a clinical trial is the processing of the massive amounts of clinical data that are generated and brought into the sponsor for assimilation, analysis, and reporting. In the case of the COVID vaccine study, simply looking at the inflection point where we executed the primary analysis based on having 90 positive cases as defined by the protocol. At that point alone, we already had collected 105 million data points in a 4-month window. And within all of those case books from all those subjects, there were 46,000 subjects, 1 million free text phrases that were inserted by study coordinators, nurses, and physicians throughout the case books that was generated in the trial. And even that free text is critical because there could be hidden unreported adverse events, concomitant medications, other issues that we have to have individuals manually reading through every one of those to interpret whether or not there was a missing adverse event or other detail that should have been collected in the form of data and not text. So 6 months prior to this, we had initiated a project. Go to the next slide. We had initiated a project using this use case to find a way that to leverage a machine learning algorithm to predict data anomalies in our data. It's a part of the clinical development process that, unless you're directly involved, most people are not familiar with. But in generating these millions of data points, there are a lot of erroneous data reported that we need to go back to the clinic, the clinician, the nurse, and query them to resolve a data anomaly. Two things that just simply contextually do not make sense. So in order to train the model to detect these, we leveraged a massive amount of historical data that we have access to. In this context, it was about 400 million data points generated from clinical trials, and about 100,000 queries and responses that were issued to clinics to resolve those data anomalies. In addition to using that to train the algorithm, we also knew there were things that were very clear and direct and didn't necessarily require an AI algorithm, but could be more prescribed. An example here is where and it's a very common one where we're looking for concomitant medications that are reported in clinical trials that may suggest that an AE was not reported. For someone, a simple example of acetaminophen, there's no clear AE associated to acetaminophen, and we would inquire why. And in some cases, in a vaccine study especially, that could result in a new adverse event that wasn't previously reported of reactogenicity, injection site reaction, and so on. So the technology that we designed actually looks up approved labels for different drugs that may have been reported in the common medication section of the study, and then prompts users to assess if there's a possibility that something was not reported to justify that concomitant medication, and vice versa. An AE that should have had a concomitant medication but did not. If we go to the next slide. So the way we approach this actually has been the most significant part of the learnings, because we have applied this methodology to predicting protocol deviations, automation of coding of medical terms and WHO drugs. Even now, looking at the potential of this kind of capability to identify the conditions that can predict a safety signal. So in this example, we called it a hackathon involving internal and external teams, where we allowed teams to develop the models, train the models with this very large amount of historical data, and then provide them with a new study that was being conducted to see if they could predict the same errors that humans had predicted in the same studies. And and in full transparency, this was done in partnership with an organization called SAMA. And we were able to predict about 50% of the anomalies that the humans were able to predict with all the tools at their disposal. Very sophisticated algorithms, reporting tools, and even direct listing reviews. And once we reached that 50%, the winner had achieved that 50% accuracy in their predictions, and then we took that version of the algorithm and initiated a human-in-the-loop learning process. These original phases were without a human-in-the-loop or unsupervised learnings. From there, we were able to take those algorithms, apply them to a number of ongoing trials where humans would execute their work in the traditional means, and then review the outputs from this technology and give it feedback. And the feedback was very simple. It was in the form of a thumbs up, where the prediction was accurate, or if you will, consistent with the human prediction. A thumbs down if it was incorrect, and there was an option of a sideways thumb for scenarios where the anomaly detection was correct, but the predictive query that should be issued was not using a clear human question that a doctor or a nurse could appropriately respond to. And a big factor here, because of the context of a clinical trial, was the avoidance of false negatives. False positive may mean a little more work for someone in reviewing the output, but a false negative means that you missed a potentially anomalous data point that will be more troublesome downfield. So we intentionally had to leave the algorithms, had to allow for more false positives to avoid having any false negatives. If you go to the next slide, please. So the timeline that was set for this program was quite challenging, quite complex, and another aspect that allowed the acceleration, besides some of this technology, was the fundamental design of the protocol itself. It was a dynamic design, starting with in the same protocol, starting with a initial phase to select the optimal sequence that would progress into phase 2, phase 2 to identify the optimal dose, and phase 3 that would be powered adequately to demonstrate safety and efficacy in a large population, in this case, 46,000 subjects. And as you can see the timeline here from the phase 1 start, May, until the submission to the agency in November. So it was by far the fastest time frame we have ever worked in. And it wasn't just simply the technology, it was the open line of communications between the company and the regulators, and very open line of communications between the different functions within the organization, and of course the technology. You go to the next slide, please. So at a very large level summary, 46,000 subjects, 154 investigators, 1,000 internal colleagues that were working on this across all the different divisions and departments, focused on bringing one vaccine to the market. And the important part to note here is how much time is beyond the science, right? From when you start dosing a subject until the endpoint of a study, one could argue, is the execution of the science. Any of the time leading up to being ready to dose that first subject, and I'm not talking about the research aspects of designing the vaccine, but more the operational aspects, as well as the components from when you have that last subject, last visit, or your primary endpoint, until you're able to summarize the results. Those are very large amounts of time during which whatever you're trying to treat is still occurring, whether it's in this context of pandemic or in many other chronic cases. You're preventing that therapy from reaching the ultimate customer. So our battle cry in the development organization is every minute counts, and especially those overhead minutes where you're really just executing process and red tape versus the science of the protocol, and our primary targets to reduce. So if you go to the next slide, please. So as I said, in this scenario, our goal was to minimize time, maximize quality, and in that traditional speed, quality, cost triangle, willing to accept the cost without willing to accept any compromises on time or on quality. The fundamental exchange of information between the organization and the regulators, our ability through this technology and the process to deliver weekly data monitoring committee reviews on the data really had a huge impact. Next slide, please. So these are all the different teams and organizations that participated or contributed to the project. One apology for one error: the safety group is listed twice. This is one of those circles should read legal, because this kind of efforts do require attention to privacy and legal concerns, because when you're using data in the context of training models, you have to assure that you have the permission from the data that those individuals belongs to use for this purpose. If you go to the next slide, please. So this across the board, this reflects the amount of time that was reduced in each of the core components in executing the clinical trial, from the randomization systems timeline was reduced by 70%. The protocol design and finalization reduced by 75%. The setup of the patient-reported outcome devices, these are the devices where the subjects report back to us things like reactogenicity or QOL measures, 75%. The timeline to submit the IND, 80%. The setup and initiation of all of our clinics around the world that participated, 85%. And the database activation setup, which my group is responsible for, was reduced by 90%. The next slide, please. So the end outcome of this was from the point that a clinical center entered a data point into the data collection tool for the study, until the time that that piece of information was validated, queried if need be, and frozen, went down from 25.4 days to 1.7 days. And this was during the execution of the study. In the last days of the study, as we approached the target number of positive cases, that number was below 1. And because of this, when we reached that 90th case and we proceeded to finalize the database and make it available to our statistics group for analysis, that time window, the industry median is about 1.5 months. The best in class industry performance in this context is about 4 weeks. In this trial, it was 1 day. And it was 1 day because of all of these factors, not only the technology, but all the factors that I mentioned. But there was a huge amount of data review and reconciliation that the technology was able to basically support the clinical data manager in resolving and finalizing, that otherwise would not be possible without a technology like this. Go to the final slide, please. So some of the components that both were leveraged to make the technology efficient, but to also make the humans efficient, are things like standards, both in the design of the study and in the design of data collection tools and in the design of the outputs that were used to generate the tables, listings, and figures for the CSRs and the submission. And really leveraging our regulatory engagement opportunities to keep open lines of communication and designing the program. For this specific tool, I visited the agency on 2 different occasions to go over our plans and our approach and validation plans, and we've archived every single learning cycle of data to be able to demonstrate why the tool is giving the outputs that it's giving, and not treat it as a black box. And with that, I'll conclude my presentation. Thank you.
Thank you very much, Mr. Zombas. Very fascinating panel. Clearly, many applications of artificial intelligence across all of the steps of the process, and the regulatory piece is certainly a critical one as well. We're a bit over on time, but I think it's worthwhile that we open up for 1 to 2 questions or comments. Daniel Hoft.
Thanks, Bob. And thanks to the panel. That really was a great set of presentations. My question is, I guess, following up on, I think, something that Dr. Matthews said about the AI analysis is only as good as the data you put in. And I'm wondering about specifically with immunome, immune profiling, when you're trying to look at human immune responses and what's predictive of protection and what vaccines predict that they can induce them. There's a lot of variables there that have to probably be corrected for and considered. And so my kind of simple question is, is there any basic guidance you can give us in terms of what size of sample size that you need to be able to have meaningful predictions for a population that you're trying to model for in AI? And maybe Dr. Schenkelberg and Dr. Golahar, based upon what you presented, might be the people I'd like to hear from first, but anyone that has any ideas would be great.
Yeah, there's a number of things, questions embedded in there. I mean, there's variation. So I think at least we saw, in terms of looking at the immunome, which we described as all the components and linkages of the immune system, is these longitudinal studies over time of specific populations where you can do an intervention. You sample before at a baseline, you vaccinate, and then or it could be an infection too. You could do a CHIM study. And then you look at changes both biologically as well as clinically in terms of outcomes. You have variation over times in the samples, you have variations between individuals. And so there's becoming more and more ways, I didn't show the papers, but where you can correct with some of that, particularly through single cell analysis, combining with deep learning, where you can limit some of these batch variations. The cost of our studies were up to 120,000 per participant, which is way too much. Those costs are coming down. And we really struggled with like what size you need and what are the minimal number of samples. The early studies, I think you got to sample more to start getting an indication of where you look at. And then you would build, you would start to build in, eliminate assays that you don't need. And you kind of start with a general, like somewhat hypothesis neutral in terms of where you're sampling, identify where you see signatures, and then expand studies. And I think you really have to work on, efficiency, cost controls. This is not, these are really initial studies that are giving you initial like insights, but not like core predictive signatures. And I think over the next 5 to 10 years, we'll get better, things will get cheaper, and we'll start to know where to look at. Does that start to answer your question?
Yes, and yes, it does. Thanks.
Yeah.
I'll just add that, the serology portion is a little bit easier to do high throughput. It's the T cell side of the house and the durability that we often don't find out as a problem until many years later, or when something is reintroduced and challenged. So I can't give you a number, but we should invest a lot more in understanding both the B cell, T cell side, as well as serology and what is impacting long-lived durable protection.
Thank you.
Well, again, I want to thank all the members of our panel. You've given us a lot of things to think about around the AI spectrum. And we'll now turn to our next panel. Our next panel is Innovative Approaches to Improve Adult Immunization. The COVID-19 pandemic provided many lessons as well as a stark reminder of the importance of equitably vaccinating people across the lifespan. Vaccines play a key role in healthy aging, protect those who may have a higher chance of serious illness or vaccine-preventable disease. In this session, we'll review data on the impact COVID-19 has played on some routine vaccines for adults generally, as well as those specifically recommended for pregnant people. We'll also explore innovative policy and legislative options and post-pandemic innovations to address systemic issues in delivery in long-term care settings. Our first presenter is Nandini Selvam from IQVIA, followed by Markeisha Jones from the Center for American Progress, and then Elizabeth Sopcich from AMDA, Society of Post-Acute and Long-Term Care Medicine. Ms. Selvam, I see you on the, and your slides are up. You have the floor.
Thank you. Thank you for the opportunity to present. So as I was just introduced, I'm going to be talking about adult and maternal vaccination trends in the US. Next slide, please. So we actually took this on, and I'll explain a little bit about the data, but to really understand the impact of COVID on routine adult and maternal immunizations, specifically influenza, pneumococcal, shingles, and then Tdap in the maternal setting. We use patient-level data representing both private as well as public data sources and insurers across all 50 states of the US. What this really means is we used administrative claims data that we have access to in all 50 states of the US. It's a fairly large population, so we started with approximately 260 million adults, and we defined that as 18 plus. And when we were done with inclusion and exclusion criteria, it left us with about 60 million for tracking. And vaccination rate was calculated as the number of adults who received each vaccine out of eligible adults, and it was aligned with the US population. So this is really generalizable and representative of the US population. I'm going to actually skip the key findings and insights here because I'll go through it in the subsequent slides. So can we move on to the next slide, please? So this gives you a good bird's eye view of the summary of across each of these vaccines. So when you look at this, influenza as you can see, the green line is for the 65 plus. Across the board, what you're seeing is, at least in flu, in the last couple of years, we saw a peak in the 20 to 21 period, and then it's declined slightly in the older age groups, and then a little bit more in the younger age groups. Later on, after I finish up this review of this particular set of data, I do have a more supply chain level data view that I can also present. When we look at shingles, we're looking at, this is not an annual vaccine, right? So this is only for the 50 plus. And so when you look at this rate, it may look smaller than you've seen in other settings, but that's because we're only looking at the eligible 50 plus year olds. So if they're already vaccinated, or if it's ongoing, then we don't see them in the denominator of this population. Pneumococcal was interesting. So it did improve in the last couple seasons, last couple of years. And I think that's primarily because of shared decision making, but also because of supply chain issues being resolved. And finally, we look at Tdap in pregnancy, and what we're seeing is it's pretty much hovering in the same spot over the last couple of years. And it's just about 50%, depending on how it's broken out. And we look at this amongst 18 to 49-year-old women and based on year of delivery. Next slide, please. So each of my subsequent slides about the vaccines, the individual vaccines, is going to be oriented this way. So you'll see it by age group, by race, ethnicity, in a fairly crude fashion. So you're looking at Asian and others, white, Hispanic, and Black, payer channel, urban, rural status. And I do have a big asterisk for this particular field because only about 5% of our cohort had that status indicated. So this is a very small group. So all these shifts you see, and I know that in this setting, it seems like rural is better than urban, and I've been asked about it because it's different than other national averages. So that is my caveat for this particular strata, as well as household income. And when you look at flu, what you see is that there were declines, and it's pretty obvious across the different age groups, across the different race ethnicity groups. What you see here is that it's much better among Asians versus the other race ethnicities, followed by white, and then Hispanic, and then Black. And then the payer channel, it's better in the public versus the private. And then urban rural, as I just said, is sort of a set aside. And then you have the highest income groups actually being more adherent and taking influenza vaccines versus the others. Next slide, please. This is actually quite a neat view. What it does is it gives you a geographical perspective of how this looks. The map on the left is for the 18 plus age group, and the right is 65 plus. And what you see is the vaccination rates compared to, so it's October 22 through September 23, compared to the same period the prior year. Anything that's red, and the darkest reds show you sort of the worst declines versus greens where you've seen improvement in flu vaccine uptake. And what you see that in the 18 plus, there were decreases in about 39 states versus in the 65 plus age group, about 20 states versus a much larger population in the younger age group. Next. We're looking at shingles here, and it does seem like it's improving in the different strata as well as just over the years. And we're still, there are a number of reasons for it, but what this really does tell you, though, is still there are many opportunities to improve vaccination across the board. So while this is an improvement, I think there's still a large gap in how much we should be covering with vaccinations. Next slide, please. Same thing here. So you're looking at the 50 plus on the left, 65 plus on the right. And what you're seeing in 22 states in the 50 plus, there were improvements, but then decreased in about 48 states for the 65 plus, in looking at the 22 to 23 versus the 2019 data. I know I'm flying through this, but I know we're behind, so I'm trying to keep on time, and I'm happy to take questions for more details. Next slide, please. So this is looking at pneumococcal, And the recommendations, just for reminder, is 50 plus for Pneumovax, 65 plus for Prevnar. Here we have seen sharp increases. And again, this is, we believe, as a result of shared decision-making as well as supply chain issues being resolved. So we're hoping that this will be a continued trend up for pneumococcal. But again, the Black and the white populations seem to be the least vaccinated as compared to the other populations we've looked at. Next slide, please. And then we're looking at this for the 65 plus age group versus the 50 in the prior slide. Again, similar sorts of trends, same sorts of reasons we believe in terms of just improvement. But again, you know, I think my overarching thought and sort of thought process on this is there's still a lot that can be done. I think with more improved programs, I think a programmatic approach, grassroots effort, perhaps there's definitely lots of room for improvement in terms of vaccine uptake, routine immunization uptake. Next slide, please. So you can see here, there's a lot of green. There are just a couple states with some red, but the vast majority of the country, it looks like it's improving. Couple really red states, and then there are some that are pink in the 65 plus group. So there's some declines, but overall, it seems to be a better story for pneumococcal than some others. Next slide, please. And so this is looking at Tdap in pregnancy. And then again, when we look at it by age group, it does seem like the older population, the 35 to 49-year-old women, are more likely to get Tdap versus the younger ones. When you look at the 18 to 24, it's the lowest vaccination rate, but even this, right? So we look at this and we say, wow, this is great. I mean, women are getting it, but not really. So if it's 50%, it hovers about 48% when you look at it combined as a cumulative look. That's still more than half the population of women not getting vaccinated. From a race ethnicity perspective, Asian seems to be higher than white versus Hispanic, and then followed by the Black population. And here we see that privately insured patients are higher than the public insured. Urban, rural, same caveat. And then household income, again, the highest to lowest is what we're seeing here. Next slide, please. And then you'll see some grayed out areas in this particular graph or map, sorry. That's because we didn't think we had sufficient volume in those states to make a real decision on how the trends were going. So we actually excluded those states from our analysis. And then in the rest of the states, what you're seeing is that overall, the vaccination rate has improved in 28 of the states when compared to 2019 for the 18 to 49 year age group. I believe that's my last slide. Is there another slide? Okay. So before I actually stop talking about this, one of the other things I wanted to do, which there is no slide for, is to present a view of sort of big picture, really near time results on what's happening with this current season. So we have what's called the National Prescription Audit, which is a supply chain patient level, but looks at the sell out data from pharmacies. So when we looked at the 2024 season versus the exact same time in 2023, right? So it's from August 22 to February 3rd, 2023. And then again, looked at August 2023 to February 3rd, 2024. We looked at 6 different vaccines. So I looked at COVID, which is something that we haven't talked about here yet, but I thought you might be interested in seeing. In the 24 season, we had 31.1 million individuals who did get COVID vaccines versus 42.3 in the prior year. And so there was a decline of 27% in COVID vaccinations. Influenza, there was a decline of 11% from the prior year. So it's the same exact timing in terms of what's considered a season. Tdap, there was actually an improvement of 13%. Shingles, a 1% increase. Pneumococcal saw a 37% increase. And then RSV, it's just we've got 9.6 million as the number of individuals who have taken RSV vaccine in this since it's been launched. So this is just some extra information that's out there that I wanted to present to this committee. And if there are any questions, I'm happy to take it, but thank you for the opportunity.
Thank you very much, Ms. Salva. We'll turn to the next presenter, Markeisha Jones from the Center for American Progress. Ms. Jones, your slides are up. I see your face on the line.
Great, thank you. Before I get started, just to give a kind of brief introduction about the Center for American Progress, or CAP as we like to say, in case folks don't know. CAP is a DC-based policy, think, and action tank. So we create and advocate for progressive policy ideas through our research and by working with legislators and those in the administration to push forth those ideas. Very brief, high-level summary of the organization, just to kind of ground everyone. So today I'd like to talk about some work that we have done on adult vaccine access, specifically around some advocacy we've done on a coverage program for uninsured adults. If we could go to the next slide. Great. So just to kind of set the scene for vaccine coverage in the US. So thinking about the different groups that have coverage for vaccines, so private insurance, the ACA has said it so most folks under private insurance have coverage for recommended routine vaccines without any cost sharing. With the passage of the IRA, we now have closed a lot of the critical cost sharing gaps within Medicaid and within Medicare, brought a lot of equity to vaccine coverage in these programs, eliminated the cost sharing for adult vaccines under Medicare Part D, and then also made it so the folks who were in the non-expansion population in Medicaid were able to get vaccines without cost sharing. And so the IRA brought, like I said, IRA brought a lot of equity to vaccine coverage in those programs. So those groups have coverage. And then obviously vaccine for children, like most people know, very, very significant in changing the tide for childhood vaccine rates, covers almost half US children for getting their routine vaccines, and a major reason why we have high vaccine rates, the high vaccine rates we have today for childhood vaccines, although there is still quite a bit of work to be done on the childhood vaccine forefront. So this leaves about 24 million non-elderly uninsured adults without comprehensive, no-cost vaccine access. This is a significant population who don't have access to vaccines without any cost sharing. The Biden administration has included in both their 2023 and their 2024 budget proposals implementing a program called Vaccine for Adults, which would be modeled after Vaccine for Children's, that would establish a program that would cover vaccines for uninsured adults, routine vaccines for uninsured adults. If we could go to the next slide. Great. And I'll breeze past this slide pretty quickly since my co-panelist was able to give you guys some really great updates on vaccine rates among adults. But one thing that I just want to note, since I am focusing specifically on uninsured adults with this presentation, they pointed out quite a number of disparities in different subgroups, whether racial, geographic, things like that. But I just want to note we would expect a lot of gaps in rates to be even bigger with uninsured folks because cost would be an additional barrier. So we could go to the next slide. And then this is, you guys already know this as physicians and vaccine advocates. So these stats are not new to you, but I felt it important to emphasize how significant the impact of vaccine preventable diseases is in society, causing many preventable diseases and many preventable deaths, and costing society billions of dollars each year. So a pretty big, significant problem that we need to be moving the needle on. Next slide. Okay, so this is where I want to get into the program. And so currently what we have established is Section 317, which is a fixed discretionary funding program. So this means that it can get cut each year. The funding levels are flexible and can fluctuate. And actually estimates from the CDC show that it's already severely underfunded based on the many uses that it already has. But this program is already in place. It is meant to cover public health infrastructure needs, which if you see this list, this is only a short list of some of the some of the different funding uses for the program, but covering vaccine education and communication, covering data systems, administration, distribution, outbreak monitoring and response, research on vaccines and safety and effectiveness, and then also some very limited uninsured adult vaccine purchasing. So it is covering a very wide array of different uses. And so because of this, very little of that Section 317 funding is going towards uninsured adult vaccine purchase. In fact, estimates are showing less than 5% is actually going towards uninsured vaccine purchase, which is really setting it so it's a limited amount of vaccines that are actually able to be purchased and accessible for the uninsured population. So alternatively, a vaccine for adults program, which it would be dedicated solely to the purchasing of uninsured adult vaccines. It would be much more expansive, allowing us to purchase a wider variety of vaccines, a larger supply of vaccines, and making that so it's that wide variety and that larger supply is available in every jurisdiction, but then also making it so that we can do advanced contracting with manufacturers as well. Vaccine for adults would also help facilitate expanding provider networks, making sure that we're able to capture the different care settings that are more regularly accessed by adults. And so thinking about urgent care facilities, pharmacies, things like that. But then most importantly, this would be a mandatory funding model. And so funding would be consistent and it could be relied on to serve the needs of uninsured adults. And so we could make sure that everyone has access. Next slide. And this is where I want to spend quite a bit of time, kind of setting the scene for the political landscape for vaccine for adults. And so that's probably the question many folks have. What is the political reality for this? So like I mentioned earlier, the Biden administration has included this in their budget request for the last couple of years. So this signifies its importance to the administration, but of course, congressional action is needed. And so there are several congressional champions for this work. And there is, in fact, draft legislation in development. CAP has provided some comment on that. It just has not been introduced or released publicly. And so that is still in development. But one of the big things that we're consistently hearing of what's hindering the progress in Congress on this is folks don't understand why we need vaccine for adults. The question is, we have 317. Isn't 317 already doing this? And one thing that we're trying to do is make the message clear that 317 is not, in fact, already doing this for some of those reasons that I already outlined for you guys in the last slide. The fact that it's the discretionary funding, so it's limited funding as well. Those funding levels don't meet the needs of the needs of the community, especially it doesn't meet the needs given the fact that the funding has so many different uses involved in it, being vaccine education, research, all of those things beyond just the purchasing of the vaccine. And then also the fact that, the different levers and authority within Section 317 doesn't allow for it to capture those non-traditional providers, such as, like I mentioned, urgent care pharmacies, things like that. And so there are some key distinctions between a vaccine for adults program and 317 that makes it so that we need VFA. And so that message needs to be dispersed throughout Congress, but it is such a nuanced and difficult message to describe. And so we're trying to help make that message as clear as possible. We even put together a congressional memo explaining the differences side by side and distributed that out to folks, but more work is needed there. One of the other things that is really holding up progress on this is just a general lack of appetite for vaccine work. I mean, we've all seen the rise in anti-vaccine rhetoric, both in the public, but also in the legislature. And so that is really, there's not really a lot of appetite for movement on this type of work. But then beyond that, as I mentioned, this would be a mandatory spending program, which is central to this being successful. We need consistent funding for this to be successful. But there's also not appetite for more mandatory spending. We're already seeing the difficulty to getting a full year spending package passed in Congress. We've been seeing that fight play out for a while. And so another mandatory spending program, there's not a ton of appetite for that as well. And it would need to be, this is the kind of program that would need to be attached to another moving policy vehicle. There was hopes that it would be attached to vehicles such as the Pandemic All Hazard Preparedness Act. That has not panned out, but it would need to be attached to another major moving policy vehicle. And at the current moment, there is not one that is ideal. And so that's another issue. But then beyond that, and we all know this, there's just generally in public and then also in policy making, there's a lack of appreciation for preventative services, especially for legislators who are making the hard and difficult decisions on funding. They're deciding on funding decisions between programs, pulling money from one budget to the next. It's hard for them to really grasp the long-term effects of preventative services, but also the wide-reaching effects of preventative services, that spending money on vaccines not only saves us money in the long run, but it saves us money in other parts of the budget in ways that's just not always so easy to quantify. And so that is another challenge towards communicating the message on why we need this and why it's critical. And then the last thing I'll say on here is also there's a bit of narrative work, quite a bit of narrative work that we need to do on rebuilding trust and authority for public health and also for the CDC as an institution. We've all seen the congressional hearings and the backlash following the pandemic. And because VFA would obviously be a CDC program, we need to, in order to gain congressional support for the program, we also need to rebuild CDC authority and support. And so that's kind of a lay of the land of the political landscape for VFA. We continue to push forward, but need additional support and need to continue to build champions. There are a couple of different ways I think providers can be especially impactful with building narrative and explaining why this program is needed. Providers are trusted and expert messengers, so it's important that you're integrated into policy advocacy efforts to expand vaccine access. Using patient stories are extremely powerful. Explaining the impact of preventative services, explaining why the current system isn't working is important. But then something else that I also want to make sure that I want to be clear about is that vaccine for adults is only one part of a larger strategy to boost adult vaccine rates, because cost is only one barrier to vaccine uptick. While having insurance coverage removes the financial barriers, there are other barriers that still exist. Most importantly, vaccine education, which is what I mentioned earlier. So Section 317 tries to address that. And so I have in a little on the side here a couple of different policy options that need to be done in concert with establishing vaccine for adults. And so expanding 317 funding would allow for more improvements in vaccine education. Also, we need to address vaccine mis and disinformation, working in partnership with social media companies. And then I'd be remiss to not mention the CDC Bridge Access Program, which is covering COVID vaccines for the uninsured population through the end of the year. The program is serving as an important opportunity to test some of those key mechanisms of a future VFA program, such as working with pharmacies. That would be really impactful for making sure we get access to vaccines where people actually need them and would be able to get them. And so that concludes my presentation. I will hold for questions at the end, but that's it. Thank you.
Thank you very much, Ms. Johns. Our final presenter of this panel is Elizabeth Sobchak from AMDA, the Society for Post-Acute and Long-Term Care Medicine. Elizabeth, your slides are up. I see you on the line.
Thank you. It's great to be with you all this afternoon. Next slide, please. What I'd like to do today is provide some context for immunization in a long-term care setting. Over the last few years, we've seen how important it is to protect both the residents and the staff in that setting. I'd like to share a project overview of our Moving Needles project, the findings and progress that we've made, specifically our quality improvement pilots, our frontline staff survey, and some EHR-IIS interoperability efforts. And finally, I'd like to identify some key opportunities for improving rates among both staff and residents. Next slide, please. So it's really critical to understand the environment that we work in long-term care. It is one of the most heavily regulated industries, and there are different regulations for skilled nursing facilities, assisted living facilities, and home-based care. So you need to know which type of facility you're working in to understand if you're at federal or state level legislation or regulation, and which part of CMS you're working under for those regulations. As you all know, there is very short staffing, generally low-wage workers who are working with high-need residents. The shortage across the healthcare system certainly exists, and it's exacerbated in long-term care specifically. Those who do stay are burned out more quickly, and so we have very high turnover rates. We also have more complex resident needs. The individuals that are in assisted living now are ones who used to be in skilled nursing facilities. The ones who are in skilled nursing facilities currently have a higher level of care need than we have seen in the past. The other piece that's helpful to understand is that we have a number of real estate investment trusts that are purchasing buildings, and so it's no longer the clinical component that's driving the decision-making. Profit margins are very slim, and often as you're paying others to run the building and lease the building from them, those financial dynamics have changed. And then finally, immunizations are dependent on leaders championing and setting the vision, as well as directors of nursing and/or infection preventionists executing amidst many other immediate job needs. The challenges that they're dealing with are extensive. The clinical topics that they're dealing with are extensive. And so really having a champion and a vision can make a big difference. Next slide, please. To level set a little bit with AMDA, we are the only medical specialty society representing the community of medical directors, physicians, nurse practitioners, physician assistants, and other practitioners working in various post-acute and long-term care settings. We have about 3,500 members currently. We have a board that offers a certificate of medical direction, and we received the Moving Needles cooperative agreement in fall of 2021, which is what I'd like to talk about next. Next slide, please. Next slide. The goal of this 5-year cooperative agreement from the CDC is to make routine adult immunizations a standard of care for post-acute and long-term care residents, as well as an expectation for employees. We have several components that we're focused on through the cooperative agreement, both directly with the facilities through quality improvement programs and addressing things at a more systematic level for all facilities. For example, integrating routine immunization reporting to state IISs. We'll also be working on a cost-benefit analysis very shortly. Next slide, please. Next slide. So we are in the second round of 2 rounds of quality improvement pilots. We have the final data from our first round. These are the rates for the average vaccination in all 9 of the facilities that participated in the first round. We have an upward trend for all vaccination rates during the period of the project, even for Tdap and shingles. Not all of our facilities focused on those, and so what you see is really representative of only the ones that did, and we still made a fairly significant improvement. You see the zeroing out of rates in September for both COVID and flu. We had a new bivalent booster when we did this round of the pilots, and then a monovalent booster in the second round, and we reset the rates in September for influenza as well to reflect the new season. Next slide. In many facilities, our COVID bivalent booster rates reached the same or higher than the facility's primary series rates at the start of the pilot. In almost every facility, our influenza vaccination rates increased, and in many facilities, our pneumococcal vaccination rates were significantly higher than at the start of the pilot as well. So what worked? How did we get there? The facilities implemented structured processes and procedures because of the pilot. They routinized their offerings and they expanded what vaccines they provided. I can't say how foundational that is to improving rates. They literally offered Tdap and shingles and residents said, yes, we'd like them, and the rates went up. But that's not the case in all facilities right now, and just expanding the offerings could do a lot to increase rates. They also checked the status on admission or used reminder recall systems. Having a renewable consent document for multiple vaccines on admission saves a lot of time and energy, and we've seen a lot of success with that strategy as well. A lot of our sites organized vaccine availability outside of clinic times for their residents. Increasing that accessibility led to a direct increase in rates. They assigned someone or a team to be responsible for the process. Nothing new there. Having a champion makes a difference. And they used the state IIS to get data on resident history. Next slide. We did have some pain points too. The facility billing during the Part A stay for Medicare was challenging once the Public Health Emergency Act was over. Pharmacies were able to directly bill Medicare on behalf of facilities during the public health emergency, and now the facilities must bill directly. It is cumbersome for them to do so, and it's been challenging for them to offer vaccine to Part A stay residents specifically. There was confusion around billing procedures for Part D vaccines. Finding histories without an IIS was difficult, and getting consent from family members for residents who were unable to assent for themselves was challenging. Next slide. We developed a billing guide that we just recently released to help clarify for facilities and for pharmacies who needs to be billing and how they need to be billing by type of Medicare, Part B or D vaccine, as well as whether the resident is in their Part A stay or a long-term stay. There are a variety of complexities. It's incredibly hard to navigate. And so this is just one step to try to make it a little more clear about who can bill for what. Next slide. Staff were challenging, more challenging than the residents for sure, but we did see an average trend upward for flu and a slight upward trend for COVID and hepatitis B as well during the first round of our pilot. Next slide. All of our facilities struggled with the bivalent booster rates. Vaccine fatigue spilled over to influenza in some facilities. We saw that strategies really needed to be tailored to individual circumstances. And so the successes occurred when facilities made vaccine more accessible, when facilities addressed staff in cohorts. They would take the kitchen staff or the housekeeping staff or the CNAs and offer education in cohorts. And when they persistently offered the vaccine, it couldn't just come in once for a clinic and be done. What also worked was identifying the reason for the lack of vaccination. Sometimes, frequently, it was a lack of convenient time or location. We saw that offering it 3 times from a trusted peer or staff person drove rates. And there was also still the more traditional hesitancy. But separating out those issues really helped identify what strategies would work to improve rates. We also had a number of facilities step back if the continued offering of vaccine pushed staff further away from the mark of getting them vaccinated. We asked them to focus on building trust, not specific to vaccines, but just between leadership and frontline staff. And again, making vaccines accessible and providing reasons for the staff and being able to get their records, to bring their records in if they're vaccinated outside of the facility. We also found that incentives worked only around the concepts of community and camaraderie building. If you were working towards incentives that built your culture of vaccination, they were much more successful than a single gift card. Even large incentives did not work if they weren't tied into building the culture of vaccination. Next slide. Data collection for staff was particularly challenging, especially around hepatitis B vaccine. There's just not a lot of tracking of data around this right now in a systematic way. And so aggregating that information for the project or for just knowing what your staff rates are was really challenging. There was not an allowable use case. I think this is changing a little bit now, but many of our facilities could not look up staff vaccination history in the IIS, and that was a challenge. All of our facilities struggled with the COVID bivalent booster rates. That fatigue spilled over to flu. The other piece that's really important to understand is that hesitancy is reflective of the communities from which staff come. Staff are not hesitant because they are employees of long-term care. They're hesitant because they're coming from communities that are traditionally hesitant. And so only addressing the hesitancy in the workplace setting is not sufficient. The other huge challenge that we have right now is that with commercialization, facilities are unable to offer the vaccine, COVID specifically, on site. Long-term care pharmacies are considered out of network with commercial insurance, and that's who delivers vaccines to the facilities. And so without that access point, we've seen rates plummet. Next slide. We started our round 2 pilot in July of the past year. We have 4 chains participating now with 3 facilities in each chain. We have geographic diversity from the East, Midwest, South, and West. We have both skilled nursing and assisted living, as well as for-profit and nonprofit institutions that are a part of the pilot. We have a more directed process around the standards for adult immunization this time around, and a strong focus on standardization and operating procedures. Our goal ultimately is to understand what works and why, and to create a change package, likely based on stages of readiness for change. Next slide. I'd also like to talk about our frontline staff survey that we did last summer and an in-service training that we've developed to help neutralize the topic of vaccines for frontline staff in long-term care. Next slide. We did a survey to understand what types of information frontline staff would like to receive regarding immunizations, trusted sources for vaccine information, as well as preferred modalities, sources, and formats for professional development. We used the survey findings to develop a training module and a distribution plan to encourage vaccine uptake among staff. Next slide. The key takeaways of the survey are that respondents are motivated to protect themselves and others from illness. The frontline staff see the value of protecting themselves and others from getting sick. Half of the frontline staff in the survey accepted vaccination as a responsibility or a requirement for long-term care staff. But this is really the key piece here. Respondents' confidence in protection through vaccination specifically is low. And so if they don't believe in the protective aspect of vaccination, they look to other methods to protect themselves and others from getting sick. Many of the respondents view vaccination as a personal decision, and they want balanced information to make their own health decisions. They don't want what they called a sell job on just the benefits. And they want that information, no surprise, from their healthcare providers first, similar to many other surveys that have been done. Government agencies were seen as trusted sources, as well as coworkers with medical training. So for our training, respondents prefer a brief paid-in service by a direct supervisor or administrator. And so we developed an in-service slide deck and supervisor training that incorporate those findings. Next slide. Both of those are posted on our website, movingneedles.org, and are available. We also have been working towards greater electronic health record and IIS interoperability. Next slide. The goal of this work really is to have the facilities be able to view resident history and make that process much more efficient for them to recommend what a resident needs and begin that process to vaccinate them with what is needed. We have 2 documents. One is a technical mapping document with 5 keys to connectivity, a workbook for self-assessment for the EHRs, and it's based on responses and interviews with multiple long-term care EHRs. These were groups that were left out of the Meaningful Use Incentives program to connect with public health when it first came out, and so they lag behind the ambulatory care EHRs. We also have a second paper with implementation considerations that identified that sustainable funding is critical, that we need to ensure awareness and understanding of connectivity benefits to strengthen and monitor collaborative action. We need to positively incentivize connectivity, and we need to reduce the operational and technical burdens of connectivity. Both of these papers were written in concert with ERA, who was hugely helpful and ensured that we had IIS participation and input into our consensus recommendations. Next slide. And so now I'd like to talk briefly about where I think there may be some key opportunities for innovation in the long-term care space. Next slide. Thinking expansively about solutions to increase on-site accessibility, especially addressing billing challenges for residents and staff, is an absolutely key area for us to think about how to increase rates. Having on-site accessibility is what I've heard described as a 6-inch chasm. It seems like we should be able to make this happen very easily, and yet the billing challenges for both residents and staff are preventing that. Another key opportunity is providing structural support and sustained technical assistance for implementation of standard operating procedures. being able to use renewable consent documents, working towards standard operating procedures is an easy way to increase access. And once the cooperative agreement is done, there won't be a group that's providing that sustained technical assistance. Embedding leadership training for medical directors, DONs, nurse practitioners, and other clinical leaders in facilities, including how to build trust, is a key component of success for our quality improvement pilots. Where you have engaged leadership, you have success in a quality improvement pilot. Where you do not have sustained leadership, it's really challenging to have a vision, to have the facilities buy into that vision, and to spend the time that they need and take that time away from other activities that they're currently doing or added in. There's also an opportunity to focus on interactive education opportunities that address the true concerns of staff, namely perceived low vaccine efficacy from sources they trust. If we can target the messaging to be to their direct concerns, I think we've got a lot of data that we can use to support these efforts. The fifth opportunity is considering incentives to further EHR-IIS interoperability, supporting increased awareness and understanding of the benefits of connectivity, and working towards reduction of operational and technical burden here. There are a lot of technical and operational burdens to address, and we need sustained funding to be able to do so. The last place is to consider additional connections between the long-term care and immunization communities. For example, more representation at NVAC or ACIP meetings, and having more systems that are built on adult versus a pediatric infrastructure. We don't want to pit the two against each other, but building more long-term care specific expertise will benefit both the residents and the staff and the delivery systems in these facilities. Next slide. Thanks so much for the opportunity to present to you today, and I look forward to your questions.
Thank you very much, Ms. Sovcik, and I want to thank all the members of this panel. Are there any questions or comments from members of the committee? Steve Rendernecht. Go ahead, Steve. Steve, can you go ahead?
I'm sorry. Thanks much for the discussions. I enjoyed listening to that. Hey, yesterday we had a discussion and kind of a celebration of 30 years of the VFC program and the success that that has showed. So I'm really hopeful that the proposed VFA program will be similar, and we can talk about that in the future. My question about the VFA program, right now with VFC, we receive vaccine for uninsured and Medicaid. Would the proposed VFA be similar in that providing vaccines to the Office for Medicare and uninsured, or is it just uninsured? And if Medicare is included, would it be both Part A and Part D vaccines? The reason I ask, I think not being able or making it difficult to give Part D vaccines to adults is a real roadblock. And I'm sure the vaccination rate has been affected by that when it comes to giving that in the office setting and not sending to a pharmacy. So just a question maybe for Ms. Johns.
Yeah, thanks. Happy to answer that. So the current proposal is specifically for the uninsured population. And that's I'll say also the way that the proposal is framed at the current moment is that it's a capped dollar amount. And so while it is mandatory funding, it's still a limited amount of mandatory funding. So I think what I have seen the administration propose was $12 billion over 10 years, I believe. Don't quote me on that. I have to go back to the last budget proposal, but if I'm remembering off the top of my head, I think that's what it was. But it would be for the uninsured population only. And in terms of the Part D coverage, that should be addressed by the IRA. Now, the mechanics of how that's gonna all get worked out, I think it's maybe still happening, but that should all be addressed by the IRA at this point. So there should be full coverage for that without cost sharing.
Hey, thank you much.
Courtney Londo, please go ahead.
Hi, thanks to all of the panelists. This was a really interesting discussion. I just wanted to make a comment on behalf of ERA, the American Immunization Registry Association. We're really thrilled that this important work is being funded. Interoperability between long-term care, EHRs, and IAS has been an ongoing issue. It really needed to be funded a decade or more ago, but now is better than never. And just wanted to point out that the work that Elizabeth presented is one step toward getting long-term care connected to IAS. It doesn't mean that all long-term care facilities are connected today or will be tomorrow. There's a lot of work to be done, but the recipe for successfully connecting has been developed, and now the funding is just needed to continue to make those connections. So thank you to Elizabeth for contributing to this important work, and thank you to the rest of the panel.
And I want to again thank all the members of this panel. You know, we all recognize we've got a long way to go to catch up on many of our vaccination rates following the pandemic, and there are, to put a positive spin on it, plenty of opportunities to use vaccine protection for our patient populations. We are now going to take a break. I now have 2:35 p.m. Eastern Time. We will be on break until 2:45 p.m. Eastern Time. I apologize for the short break, but I want to get us back to on time. Thank you for joining us for our second day of our February 2024 NVAC meeting. We'll see you back at 2:45 Eastern.
Produced by the U.S. Department of Health and Human Services.